Dear students, in this video we are going to learn about digestive processes, absorption, assimilation and digestion. First we are going to see about the process of digestion. The process of digestion is accomplished by mechanical and chemical processes. So during digestion, the solid food is converted into absorbable and assimilable forms. So first we are going to see about digestion in the buccal cavity. The mechanical digestion starts in the mouth by grinding and chewing of food. So that is called as mastication. The smell, sight, taste and mechanical stimulation of food in the mouth triggers a reflex action. So because of the reflex action, it results in secretion of saliva from the salivary gland. So what are the components of saliva? The saliva contain water, electrolytes such as sodium ion, potassium ion, chloride ion and bicarbonates. Along with this, saliva contains salivary amylase. It is an enzyme and it is also called as ptyalin. Along with this, it also possesses antibacterial agent which is called as lysozyme, and it possesses lubricating agent that is mucus. So mucus are nothing but a glycoprotein. So the saliva contains an enzyme that is called as salivary amylase. So what is the function of this salivary amylase? It hydrolyzes about 30% of polysaccharide, that is starch, into disaccharides that is called as maltose. So the salivary amylase convert 30% of starch into a disaccharide maltose. And the mucus which is present in the saliva prepares the food for swallowing. By how it prepares the food for swallowing means it moistens the food, softens the food, lubricating and adhering the masticated food in the form of a bolus. And then the bolus is passed into pharynx. From the pharynx, it is passed into esophagus. So by a process that is called as swallowing. It is also called as deglutition. So through esophagus, the bolus passed into stomach by a successive waves of muscular contraction that is called as peristalsis. The passage of food into the stomach is controlled by esophageal sphincter. This is a sphincter, that is a small valve that present between the esophagus and the stomach. So next we are going to see about digestion in the stomach. So in the stomach, the food remains for 4 to 5 hours. And the rhythmic peristaltic movement of the stomach churns and mixes the food well with the gastric juice and make it into creamy liquid that is called as chyme. The gastric juice started to secrete when the food is in the mouth itself. And this gastric secretion is partly controlled by autonomic reflexes. The content of the gastric juice are, it possesses hydrochloric acid and proenzymes. So what are the proenzyme it possesses means pepsinogen. The word contain nogen as a suffix, then you can easily understood that particular substance is in inactive state. Okay, so here the pepsinogen is a proenzyme which is in inactive state and it gets activated into pepsin, which is an active enzyme in the presence of HCl. So after activation of the pepsinogen into pepsin, active pepsin acts upon proteins and hydrolyzes the protein into proteoses and peptones. Now we are going to see about the role of hydrochloric acid. So hydrochloric acid provides acidic medium. The pH is about 1.8, which is the optimum pH for the action of pepsin. Also, the hydrochloric acid kills bacteria and other harmful organisms which are ingested along with the food material. So also the hydrochloric acid avoids putrefaction. So now we are going to see about the role of mucus and bicarbonates. So these substance helps to lubricate the food materials in order to pass the food materials easily from stomach to intestine. And also it protects the mucosal epithelium from the eroding nature of highly acidic hydrochloric acid. So apart from these substances, the gastric juice also contain another proteolytic enzyme that is called as renin. The function of renin is this renin acts upon the milk protein casinogen in the presence of calcium ion. So thus it converts the casinogen into casein. The enzyme renin are mostly found in the gastric juice of infants, and the secretion of this enzyme gradually reduces with aging. So next we are going to see about digestion in small intestine. So when the food passes from stomach to small intestine, so at that time the small intestine receives bile juice, pancreatic juice and intestinal juice. So all these juice are released into the small intestine. So after the food received by the small intestine, the movement generated by the muscularis layer of small intestine thoroughly mix the food with various secretions in the small intestine. Thereby it facilitates the final digestion of the food material. So let us see about the action of various juice that are secreted in the small intestine. So first we are going to see about bile juice. What are the component of the bile juice? The bile juice contains bile pigments. So what are the bile pigments? Bilirubin and biliverdin. So these are the pigments which are the breakdown products of hemoglobin of dead RBCs. And then the bile juice possesses bile salt, cholesterol and phospholipids. So there is no enzyme in the bile juice. So in the bile juice the enzymes are absent. So what are the functions of bile juice? Here the bile salts present in the bile juice helps in emulsification of fats. So what do you mean by emulsification? So during emulsification of the fats, the bile salts reduce the surface tension of the fat droplets and break them into small globules. So thereby the small globules are easily available for the enzyme lipase for the further digestion of fat. So also the bile juice activates lipase. So thus the lipase, the activated lipase acts upon small globules of fat and digests the fat into less substance. So next we are going to see about pancreatic juice. So in the stomach, the protein and other substances also partly digested in the stomach. Proteins and partly digested proteins which is present in the chyme when it reaches the intestine. So these proteins and partly digested proteins are acted upon by proteolytic enzymes which is present in the pancreatic juice. So what are the enzymes present in the pancreatic juice? They are trypsinogen, chymotrypsinogen. So these are the inactive form of enzymes. So next, carboxypeptidases, pancreatic amylases, pancreatic lipases and nucleases. So these are the enzymes present in the pancreatic juice. Let us see the activity of these enzymes one by one. First we are going to see about trypsinogen. So trypsinogen is an inactive form. So it gets activated into active trypsin with the help of the enzyme enterokinase. This enterokinase is secreted by intestinal mucosa, and this active trypsin in turn acts on chymotrypsinogen and convert the chymotrypsinogen into chymotrypsin. So what is the function of trypsin? This trypsin acts upon proteins and hydrolyzes the proteins into polypeptides and peptones. What about the chymotrypsin? This chymotrypsin hydrolyzes peptide bonds which is associated with the specific amino acids. So it won't hydrolyzes all the peptide bonds. It will hydrolyze only the peptide bonds which is associated with the specific amino acids. So the next enzyme is pancreatic amylase. This pancreatic amylase acts upon glycogen and starch and hydrolyzes glycogen and starch into maltose which is a disaccharide. So next is lipase. So lipase acts upon the emulsified fat which is called as triglyceride and hydrolyzes this triglyceride into free fatty acids and monoglycerides. Again, this lipase acts upon monoglycerides and hydrolyzed that monoglyceride into fatty acids and glycerol. The last enzyme, nucleases. This nucleases acts upon the nucleic acid and convert the nucleic acid into nucleotides and nucleosides. So next we are going to see about saccus entericus. The duodenum of small intestine possesses a gland that is called as Brunner's gland. So the secretion of the Brunner's gland and the secretion of the intestinal gland constitute the intestinal juice. It is also called as saccus entericus. So the enzyme present in the saccus entericus, they acts upon the broken down product of bile and pancreatic digestion. So what are the enzymes present in the intestinal juice? They are maltase, lactase, sucrase. It is also called as invertase, and dipeptidase, lipase, and nucleosidases. So these are the enzymes present in saccus entericus or intestinal juice. So let us see the activities of these enzymes. So maltase is the enzyme that acts upon maltose and hydrolyzes the maltose into 2 molecules of glucose. And sucrase, that is invertase, they hydrolyzes sucrose into 1 molecule of glucose and 1 molecule of fructose. And lactase is the enzyme which acts upon lactose and convert the lactose into a molecule of glucose and galactose. In the case of peptidase, they acts upon dipeptides and tripeptides and convert that into amino acids. Then nucleotidase. Nucleotidase acts on nucleotides and converts the nucleotides into nucleosides and phosphoric acid. And nucleosidase is the enzyme that acts upon nucleosides and converts the nucleosides into sugar and nitrogen base. Lipase. Lipase acts upon diglycerides and monoglycerides and convert that into fatty acids and glycerol. And in the case of intestinal juice, apart from these enzymes, they possess mucus and bicarbonate ions. So these mucus and bicarbonate ions are received from pancreas. So these mucus and bicarbonate ions provide alkaline medium. The pH is about 7.8. So this is needed for the enzymatic action in the small intestine. Because of the enzymatic action in the mouth, stomach and small intestine, all the macromolecules in the food materials are completely converted into micromolecules. So here the carbohydrate is converted into monosaccharides like glucose, fructose and galactose. And the proteins are converted into amino acids and lipids are converted into fatty acids and glycerol. Thus, the simple substance thus formed or absorbed in the jejunum and ileum, and the undigested and unabsorbed substances are propelled into large intestine. So here, the activity of the gastrointestinal tract is controlled by neural system and hormones. Okay, in order to bring out proper coordination. Here, in the case of secretion of gastric juice and intestinal juice, it is stimulated by neural signals, and the hormonal control is carried out by hormones which are secreted by gastric and intestinal mucosa. So, these are the process of digestion. So, next we are going to see about absorption and assimilation of proteins, carbohydrates and fats. So, absorption. First, we are going to see about absorption. So, the end product of digestion passes through the intestinal mucosa into the blood and lymph. That is called as absorption. The absorbing unit in the small intestine is villi, which is present in ileum part of the small intestine. This villi consists of a lacteal duct in the middle, which is surrounded by fine network of blood capillaries. This is the structure of villi. So the middle part, which is the lacteals. Middle part is the lacteal, and it is surrounded by fine network of capillaries, blood capillaries. Okay. So here the process of absorption involves active transport, passive transport and facilitated transport. Let us see how the digested substances are absorbed into the blood through villi. So some amount of glucose, amino acids and electrolytes like chloride ions, they are they are absorbed into the blood by simple diffusion, and these substances are absorbed based on the concentration gradients. That means these substances first at the time of absorption, at the beginning of absorption, these substances move from higher concentration to the lower concentration. That means after digestion, okay, the substance, the glucose, amino acids and electrolytes like chloride ions are more in concentration in the lumen of the intestine and compared to blood. So it will easily diffuse from the lumen of the intestine into the blood capillaries by simple diffusion. And the substance like fructose, it cannot easily pass into the blood capillaries. So it is absorbed with the help of carrier ions like sodium ion. So this type of absorption is called as facilitated transport. And the nutrients like amino acids, glucose and electrolytes, these are absorbed into the blood against concentration gradient by active transport. And here in the case of fatty acids, glycerol and fat soluble vitamins, so these cannot be directly enter into the blood. So first it gets incorporated into small spherical water soluble droplet, that is called as micelles. And these micelles are absorbed into the intestinal mucosa. And in the intestinal mucosa, these micelles are resynthesized into protein-coated fat globules, that is called as chylomicron. Then this chylomicron is transported into lacteals. And eventually, these chylomicrons are emptied into the lymphatic duct from the lacteals. And from the lymphatic duct, these substances are released into the bloodstream. So thus the fatty acids, glycerol and fat soluble vitamins are taken into the blood. And in the case of water soluble vitamins, they are absorbed by simple diffusion or by active transport. And water is absorbed mainly depends upon osmotic gradient, that is based on our body need. Apart from small intestine, the other parts of the digestive tract are also involved in absorption. So mainly other than small intestine, mainly the absorption also takes place in mouth, stomach and large intestine. So in the case of stomach, the simple sugar, alcohol and medicines, they are absorbed in the stomach itself. Certain drugs are absorbed in mouth through the blood capillaries, which is found at the lower side of the tongue and mucosa of the mouth. And the substance like water, vitamins and some minerals and certain drugs are absorbed in large intestine. So next we are going to see about assimilation. The absorbed food material won't be directly taken into different parts of the body. First, these absorbed substances are taken to the liver by the blood and lymph through hepatic portal system. From the liver, the nutrients are transported to all the regions of the body for utilization. So when the nutrients reach the cells and tissues, they started to utilize the absorbed substances for their functions, and also they incorporate the absorbed nutrients into their protoplasm. That process is called as assimilation. So next we are going to see about digestion. The digestive waste and unabsorbed substances in the ileum enter into the large intestine. Mostly these substances contain fiber. It is commonly called as roughage. And when this roughage reaches the large intestine, it is utilized by symbiotic bacteria which is present in the large intestine. And this bacteria produces substances like vitamin K and other metabolites which is needed for our body. And all these substances are absorbed in the colon, that is large intestine, along with the water. After that, the remaining waste is then solidified into fecal matter in the rectum. Then this fecal matter initiate a neural reflex. So this reflex causes an urge or desire for its removal. So the removal of feces through the anal opening is called as defecation, and this defecation is a voluntary process and it is carried out by the peristaltic movement of the digestive tract. Okay student, next video we can see about caloric value of carbohydrate, fat and proteins, and also we are going to learn about digestive disorders. Thank you students.